Hello viewers, I am Dr. Sumati from Agricultural University, Hyderabad. Today we are going to discuss about passive diffusion, facilitated diffusion and active transport mechanisms. Objectives of this class are to enable the students to understand the need for transport of molecules and the composition, structure and functions of biological membranes. Second objective will be to acquaint you with the various facilitators of transport mechanisms. The next objective is to familiarize you with the mechanisms of active transport. Now, let us discuss in detail about the mechanisms of facilitated diffusion and active transport. As all of you are familiar with, all the cells of various tissues within a living organism acquire the molecules and ions they need from their surrounding extracellular fluids. There is a continuous traffic of molecules and ions in and out of the cell through its plasma membrane. Examples of this are glucose, sodium, calcium, etc. In eukaryotic cells, there is also transport in and out of membrane bound intracellular components such as the nucleus, endoplasmic reticulum and mitochondria. Examples of this are mRNA, ATP, calcium, etc. The term membrane refers to any of these cell membranes and the term plasma membrane refers specifically to the surface membrane. Now, contribution of cell membrane to cell function falls into two categories. Number 1, it provides barrier to the movement of molecules and ions both between the various cell organelles within the cell and between the cell and extracellular fluid. Second point, they provide scaffolding to which various cell components are anchored. The chemical composition of extracellular and intracellular fluid is quite different, which demonstrates that plasma membrane prevents free mixing of molecules on the two sides of the membrane. Thus, plasma membrane is selectively permeable. Now, biological membranes are composed of lipid, protein and carbohydrate that exist in a fluid state. The carbohydrates are attached to either lipids forming glycolipids of various classes or to proteins forming glycoproteins. The lipid and protein composition of membranes vary from cell type to cell type as well as within the various intracellular compartments that are defined by intracellular membranes. Protein concentration can range from around 20% to as much as 70% of the total mass of a particular membrane. The lipids making up the components of membranes are of three major classes that include glycerophospholipids, sphingolipids and cholesterol. Sphingolipids and glycerophospholipids constitute the largest percentage of lipid weight of biological membranes. The hydrocarbon tails of these two classes of lipids results in steric limitation to their packing such that they will form disc-like micelles. The structure of these micelles results from the interaction of the hydrophobic tails of the lipids and the exposure of the polar head groups to the aqueous environment. This orientation results in what is referred to as lipid bilayer. Biological membranes contain proteins, glycoproteins and lipoproteins. Proteins associated with membranes are of two general types, integral and peripheral proteins. Integral membrane proteins, which are also called as intrinsic proteins, are tightly bound to the membrane through hydrophobic interactions and are inserted into or penetrate the lipid bilayer. In contrast, peripheral membrane proteins, also called extrinsic proteins, are only loosely associated with the membrane either through interactions with the polar head groups of the lipids or through interactions with the integral membrane proteins. Peripheral membrane proteins are most often if not exclusively found on the cytosolic phase of the plasma membrane or the luminal surface of the subcellular organelle membranes. Now, we will see the picture of typical lipid bilayer of the plasma membrane. As you can see this, in this picture you can see different carbohydrate material which is present here, glycoprotein material which is present here. This is the extracellular fluid side of the cell membrane, and these are the cholesterol molecules. This is the integral protein molecule and this is the peripheral protein molecule. So, this is the lipid bilayer of the cell, and we have extracellular fluid and we also have the cytosolic side of the cell membrane, and these are the lipid components which are present, which form the micelle on the cell membrane. So, these are the typical components of a lipid bilayer, where you have cholesterol, integral proteins, peripheral proteins, glycolipids and glycoproteins, and all of them are going to play a major role in the total transport mechanisms of the cell. Now, next we will discuss about what are the facilitators of transport mechanisms which are present in the cell. Now, due to the predominant lipid nature of the biological membranes, as we have seen in the previous slide, biological membrane is predominantly made up of lipid materials. So, due to the predominant lipid nature of the biological membranes, many types of molecules are restricted in their ability to diffuse across a membrane. Because of the predominant lipid nature of the biological membranes, many types of molecules are restricted in their ability to diffuse across a membrane. This is a very important point which you have to remember and understand. This is especially true for charged ions, water and hydrophilic compounds. Charged ions, water and hydrophilic compounds. The barrier to membrane translocation is overcome by the presence of specialized channels and transporters. Please understand this point. The barrier to this membrane translocation is overcome by the presence of specialized channels and transporters which are present in the membrane, and they are going to play a major role in the transport mechanism. Although channels and transporters are required to move many types of molecules and compounds across membranes, some substances can pass through from one side of a membrane to the other side through a process of diffusion. The definition of a channel or a pore is that of a protein structure that facilitates the translocation of molecules or ions across the membrane through the creation of a central aqueous channel in the protein. Creation of a central aqueous channel in the protein. This central channel facilitates diffusion in both the directions depending on the direction of concentration gradient. Please understand this point. This channel facilitates diffusion in both the directions depending on the direction of the concentration gradient. Now, channel proteins do not bind or sequester the molecule or ion that is moving through the channel. Please remember this point. Channel proteins do not bind or sequester the molecule or ion that is moving through the channel. Specificity of channels for ions or molecules is a function of the size and charge of the substance. Underline this point. Specificity of a channel for ions or molecules is a function of the size and charge of the substance. So, the flow of molecules through a channel can be regulated by various mechanisms that result in opening or closing of the passage. Transporters are distinguished from channels because they mediate the movement of ions and molecules by physically binding to and moving the substance across the membrane. Transporters exhibit specificity for the molecules being transported. Transporters can also be affected by both competitive and non-competitive inhibitors. Transporters are also known as carriers, permeases, translocators, translocases and porters. So, there are different names given to the single term of transporters. Mediated transporters are classified based upon the stoichiometry of the transport process. So, there are different types of mediated transporters. Uniporters transport a single molecule at a time from one direction to the other direction through these channels. Now, next we have the second example, symporters simultaneously transport two different molecules in the same direction. Uniporters transport a single molecule at a time. Symporters simultaneously transport two different molecules in the same direction. Now, next we have antiporters transport two different molecules in opposite direction. Please understand the difference. Uniporters transport a single molecule at a time. Symporters simultaneously transport two different molecules in the same direction. Antiporters transport two different molecules in opposite direction, which is represented in this picture. Now, next we will discuss about the classification of transport mechanisms and diffusion. Okay. There are two types of transport mechanisms, one is passive transport, the other one is active transport. Now, passive transport, the driving force for this transport is the concentration gradient for the nutrient across the membrane. Passive transport can further be divided into passive diffusion and facilitated diffusion. Now, second we have active transport. Now, in active transport, it uses a transport protein, but energy is required in the form of ATP to move nutrients against a concentration gradient. So, the basic difference between passive transport and active transport is that in active transport, energy is required in the form of ATP to move nutrients against a concentration gradient. Now, we will discuss about passive transport, where we have diffusion and facilitated diffusion. Basically, diffusion is the movement of molecules from one location to another location by random thermal motion. Passive diffusion can occur through lipid bilayers. Example, most polar molecules and ionized molecules enter cells very slowly or not at all, whereas nonpolar molecules enter cells very rapidly. This is because nonpolar molecules rapidly cross the membrane by directly diffusing through the lipid bilayer. We have discussed about the lipid bilayer. So, nonpolar molecules rapidly cross the membranes by directly diffusing through the lipid bilayer. The more lipid soluble the substance is, the greater will be the number of molecules dissolved in the membrane lipids and the higher is its concentration within the membrane. Oxygen, carbon dioxide, fatty acids and the steroid hormones are examples of non-polar molecules which diffuse rapidly through the lipid portion of the membranes. Now, diffusion can also occur through protein channels. Ions such as sodium, potassium, chlorine diffuse across plasma membranes through many of the integral proteins that span the lipid bilayer. Such proteins can form channels through which ions can diffuse across the membrane. The ion channels show selectivity for the type of ions that can pass through them. This selectivity is based partially on the diameter of the channel and partially on the charged and polar surfaces of the protein that form the walls of the channel, because protein is a channel where you have different walls. So, depending on the charge of these polar surfaces, the selectivity also depends. Example, some potassium channels allow only potassium ions to pass, some sodium channels allow only sodium ions to pass, some channels are slightly less specific and allow both sodium and potassium ions to pass. See the difference here. There are some protein channels which allow only sodium to pass, there are some protein channels which allow only potassium to pass, but there are some channels which are slightly less specific. So, they allow both sodium and potassium ions to pass. So, it is depending on the selectivity which depends on the diameter also on the charge of the protein channels, the movement of molecules or ions can take place. Now, next we are going to discuss about facilitated diffusion. Facilitated diffusion of ions takes place through proteins or assembly of proteins embedded in the plasma membrane. That means through a protein or assembly of proteins, a group of proteins which are present or embedded in the plasma membrane. These transmembrane proteins form a water filled channel through which the ion can pass down its concentration gradient. The transmembrane channels that permit facilitated diffusion can be opened or closed. As we said, they form channels. So, these channels can be opened or closed. They are said to be gated because they can be opened or closed. They are said to be gated channels. One is a ligand gated channel, second one is a mechanically gated channel, third one is a voltage gated channel, fourth one is light gated channel. Now, let us discuss about these channels individually. First, we will take about the ligand gated ion channels. Many ion channels open or close in response to binding a small signaling molecule which is called as a ligand. Some ion channels are gated by extracellular ligands and some are by intracellular ligands. So, that means there are two different types of ligands. One is extracellular ligands, that means outside the cell they bind. Other one is intracellular ligands which are binding inside the cell. In both the cases, ligand is not the substance that is transported when the channel opens. We are talking about the transport of molecules. We are talking about some protein channels which open or close and they are gated. So, when we are talking about these gated channels, the ligands are not the substances which are transported through the channels, they are only helpful to open or close the gate. So, it is not the substance that is transported when the channel opens. External ligands bind to a site on the extracellular side of the channel. As we said, we have different types of ligands, external ligands and internal ligands. So, external ligands bind to a site on the extracellular side of the channel. Examples: the binding of the neurotransmitter acetylcholine. At certain synapses, open channels that admit sodium and initiate a nerve impulse or muscle contraction. Now, second example, binding of gamma aminobutyric acid, which is in short called as GABA, at certain synapses in the central nervous system, admits chloride ions into the cell and inhibits the creation of nerve impulse. Both of them go in opposite direction. One initiates the nerve impulse, the second one is inhibits the creation of nerve impulse. So, internal ligands bind to a site on the channel protein exposed to cytosols. So, external ligands bind on the site exposed to outside, internal ligands bind to a site on the channel protein exposed to the cytosol inside portion of the cell. Examples: second messengers like cyclic AMP and cyclic GMP regulate channels involved in the initiation of impulses in neurons responding to odor and light respectively. Now, let us see the example of this ligand gated channel. You can see this is a channel. This is a protein channel, there is no ligand here. You can see there is a ligand attached here. When a ligand is attached here, it opens up. This protein channel opens up. So, there is a passage of molecules into the cell. This is an external ligand which is being attached to this protein channel. So, when this ligand is attached here, the movement of ions takes place into the cell. Once the movement of ions is over, then it is closed again like this. That is why we said these channels or these transporters are gated. Gated because of the binding of a ligand on outside or inside of the cell, so that there is movement of ions or molecules which can take place. Is that point clear? Now, the second gated channel which we have seen is mechanically gated channels. Examples of these mechanically gated channels are sound waves bending the cilia like projections on the hair cells of the inner ear open up ion channels, leading to the creation of nerve impulses that the brain interprets as sound. So, this is the mechanically gated ion channel. Because the sound waves create that impact, it is mechanically opened up and we feel it as, we interpret it as a sound. Now, next we have the third example of voltage gated ion channels. In this we have the so called excitable cells like neurons and muscle cells. Some channels open or close in response to changes in the charge which is measured as volts across the plasma membrane. Example, as an impulse passes down a neuron, the reduction in the voltage opens sodium channels in the adjacent portion of the membrane. This allows the influx of sodium into the neuron and thus the continuation of the nerve impulse. Some 7,000 sodium ions pass through each channel during the brief period of about 1 millisecond. During that time only it remains open. Now, the next we will go into the mechanisms of active transport. This is the pumping of molecules or ions through a membrane against their concentration gradient. It requires a transmembrane protein called a transporter and energy. The source of this energy is ATP. The energy of ATP may be used directly or indirectly. There are many different classes of transporters that couple the hydrolysis of ATP to the transport of specific molecules. In general, these transporters are referred to as ATPases. These are so named because they are autophosphorylated by ATP during the transport process. There are different types of ATPases that function in eukaryotes. Sodium potassium ATPases is one main example. These are the most thoroughly studied ATPases. These transporters sometimes are also called as sodium potassium pumps, are involved in the transport of sodium ions out of and potassium into the cells. The extrusion of sodium ions allows cells to control their water content. In addition, each time 3 molecules of sodium ions are transported out and only 2 molecules of potassium ions are transported into the cell. So, an electrochemical gradient is established. This is the basis for electrochemical excitability of nerve cells. In fact, it is this transporter action that is a major requirement for ATP production from glucose oxidation in the central nervous system. These ATPases are composed of 2 subunits, alpha and beta subunits. Sodium and potassium ATPases also associate with a family of small single transmembrane spanning protein termed FXYD proteins. Now, if you can look at this picture, this is extracellular side of the cell, this is the intracytosolic side of the cell, this is the cell membrane. You can see this is an alpha subunit, this is a beta subunit and this is another protein which is fixed to it, FXYD protein. So, these sodium potassium ATPases have alpha beta subunits along with an FXYD protein. When ATP is bound to sodium potassium ATPases, they can bind to intracellular sodium ions. The hydrolysis of ATP results in the phosphorylation of an aspartic acid residue in the subunit of sodium potassium ATPases. The phosphorylation of this residue results in release of ADP. The phosphorylation of the pump results in a conformational change which exposes the sodium ions into the outside of the cell and they are released. The pump then binds 2 extracellular potassium ions which stimulates dephosphorylation of the alpha subunit that in turn allows the pump to bind ATP again. The dephosphorylation and ATP binding causes the pump to reorient to its original conformation and releases the potassium ions inside the cell. At this point, the pump is capable of release of sodium ions again. As you can see here, this is extracellular fluid, this is a cytosolic fluid, this is alpha subunit, this is a beta subunit, this is FXYD protein. Now, when ATP is bound to this alpha subunit of this protein sodium potassium ATPase, sodium ions are transported through this alpha subunit out. When 3 sodium ions are transported out of the cell, 2 potassium ions are transported into the cell. So, all this happens when ATP is bound to alpha subunit and exit of 3 sodium ions and inside entry of 2 potassium ions. Now, next we have hydrogen ATPases. The parietal cells of our stomach use this pump to secrete gastric juice. These cells transport protons from a concentration of about 4 into 10 to the power of minus 8 molar within the cell to a concentration of about 0.15 molar in the gastric juice, giving it a pH close to 1. Parietal cells are stuffed with mitochondria and use huge amounts of ATP as they carry out this 3 million fold concentration of protons. Now, next the last one we have calcium ATPases. A calcium ATPase is located in the plasma membrane of all eukaryotic cells. It uses the energy provided by one molecule of ATP to pump one calcium ion out of the cell. The activity of these pumps helps to maintain 20,000 fold concentration gradient of calcium between the cytosol, which is at 100 nanometers, and the extracellular fluid, which is at 20 millimolar. So, this causes increase of approximately 20,000 fold concentration in the cell. We have discussed about various transport mechanisms: diffusion, passive diffusion, facilitated diffusion and active transport. Now, we will just conclude it by saying that all cells of various tissues within a living organism acquire the molecules and ions they need from their surrounding extracellular fluid. There is a continuous traffic of molecules and ions in and out of the cell through its plasma membrane. In eukaryotic cells, there is also transport in and out of membrane bounded intracellular components such as the nucleus, endoplasmic reticulum and mitochondria. Biological membranes contain proteins, glycoproteins and lipoproteins, etc. Proteins associated with the membranes are of two general types: integral and peripheral proteins. These proteins act as channels and transporters for the translocation of molecules and ions. Hope you have enjoyed this class. See you in the next session. Thank you.